In multicolor flow cytometry experiments, different fluorochromes have different emission wavelength ranges, which can overlap and interfere with data analysis. This interference can be resolved with single positive control. So, what is single positive control? Simply put, it's a sample tube with only one fluorochrome. If your experiment uses 3 fluorochromes, like APC, PE, and FITC, you'll need 3 single positive control tubes, one for each fluorochrome. There are a few requirements for setting up single positive control. First, the sample type and fluorochrome in the single positive tube should match those in the full stain tube. Second, the sample treatment for all tubes should be consistent. For example, if the full stain tube is fixed and permeabilized, the single positive tube should also be treated the same way, even if it's just a surface marker. Third, the voltage of the instrument should remain consistent for both the single positive tubes and the full stain tube during detection. Lastly, make sure there are enough positive cells in the single positive tube. So, do you still need to set up single positive control if your target marker is highly expressed and the positive and negative cells are clearly differentiated? If your panel has only a few indicators, you might be able to adjust the compensation manually without setting up single positive control. But if your panel has many indicators, it's essential to set them up. When the expression of certain markers is low, the positive cells in the single positive tube might not be easily distinguishable. The software might struggle to correctly identify the positive cells. To solve this, you can replace the low expression marker with a high expression marker, like CD3 or CD4, that uses the same fluorochrome. When designing your flow cytometry panel, try to choose fluorochromes that have minimal spectral overlap with others to reduce the need for compensation. Typically, the compensation value shouldn't exceed 100%. If it does, it might indicate a tandem fluorochrome breakdown issue. Also, the compensation value shouldn't be negative. If it is, it means some channels are overcompensated. After automatic compensation, you can manually check if the compensation is reasonable and make fine adjustments if necessary. To better distinguish between positive and negative groups, you can first screen the corresponding cell groups using FSC and SSC, which makes compensation adjustments easier. In summary, properly setting up and using single positive control can make analyzing flow cytometry results more convenient and accurate.